What if the world's most studied genome still hides its biggest secret? In 2022, scientists opened a medieval Jewish graveyard in Erfurt, Germany. Bones that had been silent for 700 years suddenly began to speak. What they revealed shattered 2 centuries of textbook assumptions. Not folklore, not legend. Hard-coded genetic truth. Modern Ashkenazi Jews, nearly 11 million people, trace most of their ancestry back to roughly 500 individuals. A bottleneck so extreme it's like trying to build a skyscraper on 5 bricks. But the real twist isn't just how few founders there were, it's who those founders actually were. On the paternal side, the Y chromosomes, the message is loud and clear. The Middle East. Lineages still shared today with Palestinians, Druze, and Mizrahi Jews. The Levant, written unmistakably in the male line. But then comes the contradiction. The maternal DNA, the line passed only from mothers to children, doesn't point to Jerusalem, Judea, or anywhere in ancient Israel. It pulls us somewhere else entirely, into the forests and river valleys of prehistoric Europe. 4 lineages dominate: K1A1B1A, K1A9, K2A2A1, and N1B. These are not Middle Eastern. They are older than Judaism itself, carried by women who lived thousands of years before the Torah was ever written. And yet, these lineages now define nearly 40% of Ashkenazi maternal ancestry. Genetic studies by Richards, Costa, Waldman didn't just suggest this, they confirmed it. Ashkenazi Jews inherited millions of mitochondrial threads from women who never set foot in the Levant. So how did this happen? How did a people who carried Hebrew scrolls, lit Shabbat candles, and prayed toward Zion inherit their very biology from women who once worshipped stone gods under the European sky? Some point to conversion, others to intermarriage. But the timing complicates everything, because these maternal lines don't match ordinary medieval European peasants. They echo back to prehistoric hunter-gatherers, to women long vanished from recorded history. So the question isn't just scientific, it's deeply personal. Who were these women, and why were their genes chosen to live on? Because if these mothers weren't Jewish at first, what does that make their descendants? Everything circles back to one mysterious woman. A woman who never read the Torah, never spoke Hebrew, never imagined that millions would one day call her their founding mother. She never knew she was a founder, but her DNA is carried by millions today. We don't know her name. We don't know what prayers she said, or whether she prayed at all. But through the lens of ancient genetics, we know this. More than 7,500 years ago, somewhere in prehistoric Europe, a woman passed on a tiny fragment of herself that would eventually define nearly half of the Ashkenazi Jewish maternal lineage. Her genetic signature, one specific branch, of K, is one of just 4 mitochondrial lineages that dominate today's Ashkenazi mtDNA pool. Alongside K1A9, K2A2A1, N1B, and a few others, these markers were not born in the Levant. They weren't even born in the Roman Empire. These lineages trace back to Neolithic Europe, long before Judaism, before Moses, before the first stones of the Second Temple were laid. This isn't a theory. A 2013 study by Costa and colleagues, later expanded by Richards and revalidated by Waldman in 2022, confirmed it. Not only are these lineages deeply European, they're also found almost nowhere else except in Ashkenazi Jews. That means something profound happened. Not randomly, but with intent. Somewhere between the 1st and 3rd centuries CE, as Jewish merchants and migrants settled across Italy, a quiet shift began. In the heart of the Roman Empire, amid temples, synagogues, crowded streets, Jewish men began forming families with local Roman women. Conversion, very likely. Intermarriage, almost certainly. Ancient writers like Philo and Josephus describe women converting in large numbers, especially in cosmopolitan cities like Rome, Ostia, and Pompeii. By the 4th century, Roman authorities were cracking down on Jewish proselytism. It wasn't a trickle, it was a wave. These weren't faceless unions, they were transformative. They fused ancient Israelite ritual with European bloodlines, weaving European DNA into Jewish identity cell by cell, generation by generation. And yet, one contradiction refused to disappear. As the maternal lines shifted westward, the Y chromosomes, the paternal lines, didn't move. They stayed anchored to the Levant, stubbornly tied to ancient Near Eastern roots. If the mother's roots are European, why are the father's roots so consistently Levantine? That question sat unanswered for centuries, until 33 buried bodies beneath medieval Germany unlocked a chapter no one expected. In 1349, the Black Death wiped out millions. For one small community, it didn't just bring death, it froze their DNA in time. Erfurt, Germany. Under a medieval Jewish cemetery, sealed in silence for nearly 700 years, scientists found a genetic time capsule. In 2022, researchers sequenced the bones of 33 Ashkenazi Jews buried in an age of terror, pogroms, plague, and fire. What they found was shocking. By the 14th century, Ashkenazi Jews had already locked in their distinct genetic profile, complete with rare mutations that still exist today. Their mitochondrial signatures were already dominated by the same 4 European lineages. Their autosomal DNA was already a fusion of Middle Eastern roots and Southern European input. The Erfurt dead carried rare variants that modern Ashkenazi Jews still carry today, as if time and evolution had pressed pause. This is the founder effect, when a small, isolated population, cut off by war, exile, or disease, passes on its unique genetic blueprint almost unchanged. In the Ashkenazi case, genetic modeling shows that around 350 to 500 individuals survived centuries of pressure and went on to form a population of over 10 million. But survival came with a cost. Those same Erfurt genomes carried mutations linked to diseases that would later become tragically familiar: Tay-Sachs, Canavan disease, Gaucher disease, BRCA1 and BRCA2 cancer mutations. Conditions that became almost synonymous with Ashkenazi identity began with just a handful of carriers trapped inside a shrinking gene pool. Roughly 1/3 of the Erfurt remains traced back to a single woman's mitochondrial DNA, a ghost mother of millions, not myth, statistics. And it all happened during one of history's darkest moments, 1349, the peak of the Black Death, when Jews were blamed, tortured, massacred, entire communities wiped out. Blood on cobblestones, bones beneath cathedrals. Yet somehow this tiny group survived. Their DNA became a relic, a record, a genetic echo. But what if this bottleneck didn't just preserve genes? What if it erased something deeper? Maybe what vanished wasn't only genetic diversity. Maybe it was truth. One of the clearest cracks in that truth runs through a priestly caste, a caste that was supposed to be unchanged and unbroken. Because one priestly line carries a secret no one expected, its origin isn't Levantine at all. For centuries, Levites have been seen as sacred helpers, assistants in the ancient temple, guardians of Jewish ritual. Their status passed from father to son, generation after generation. The assumption was simple. If you are a Levite, your paternal line goes back to Levi, to Sinai, to the Levant. But DNA does not care about tradition. Study after study, from Behar, 2003, to others, and later Waldman, 2022, found that more than half of Ashkenazi Levites carry the Y chromosome haplogroup R1a, a lineage uncommon in the Middle East. Instead, it's strongly associated with Slavic populations, Eastern Europeans, and some Indo-Iranian groups. The contrast is sharp. Kohanim, the higher priestly caste, show strong genetic continuity with Middle Eastern lineages, especially the famous J1 Cohen modal haplotype. Their ancestry consistently points back to ancient Israel. But Levites? Their paternal line tells a different story, not rooted in Jerusalem, but in medieval Europe. Genetic models date this Ashkenazi R1a Levite cluster to roughly 1,200 years ago. The genetic signatures are so tight that researchers estimate a single European man, just one, became the founding father of this entire Levite line. Was he a convert, a survivor of war, a stranger who entered the community in a time of chaos, famine, or flight? We will never know his name, but we do know this. He was not from Judea. He was not from the original temple priesthood. Yet today, tens of thousands of Jewish men trace their sacred Levite status back to his blood. And still, across the world, in synagogues and family trees, Levite identity is repeated without question. as if it were perfectly ancient, perfectly unbroken. If the priesthood itself could be reshaped by an outsider, what else in the Ashkenazi origin story has been quietly rewritten? To find the deeper answer, we have to go further back, not to Germany, but to the Mediterranean coast, where the first spark of a new identity quietly ignited. They were called Germans, but their DNA points to Greece and Rome. For centuries, the world labeled them Ashkenazim, German Jews of the Rhineland. But their genomes tell a different story. Autosomally, looking at the full spectrum of ancestry, studies like Globetrotter and others around 2017 show that roughly 26 to 52% of Ashkenazi DNA doesn't come from Central Europe at all. It comes from Southern Europe, Italy, Greece, Iberia. And this isn't a late medieval story. This Southern European contribution entered the gene pool before the famous medieval bottleneck, before the push into Poland, Lithuania, and Eastern Europe. It dates back 25 to 50 generations, roughly 700–500 CE, when Jewish communities flourished along the Mediterranean: Rome, Thessaloniki, Provence. These weren't temporary stops. They were hubs. By the 1st century CE, tens of thousands of Jews already lived in cities like Ostia, Naples, and Carthage. Hebrew inscriptions fill the Roman catacombs. Ancient synagogues stood close to pagan temples. And it's there, in the shadow of a collapsing empire, that a new identity began to form. The DNA doesn't lie. It shows deep mixture with Southern European populations long before migration into the colder north. Long before the ghettos, long before Yiddish ballads, the German Ashkenazi story was already being written on Mediterranean bones. This isn't just about geography, it's about where identity began. The genes whisper of olives and marble, not only forests and frost. By the time these people reached the Rhineland, their southern origin was already fading from memory. They became Ashkenazi, German Jews, but their genome never forgot. How did a Mediterranean fusion become a Germanic identity? To answer that, we must stop staring at maps and start listening to language, because language carries echoes that geography can't explain. The language of exile wasn't born in Germany. It carries the ghosts of Eastern trade routes. For years, Yiddish was described like a simple recipe. A little medieval German, a pinch of Hebrew, some Aramaic on top, mixed somewhere between the Rhine and the Danube. But something didn't add up. Its grammar bends strangely eastward. Its syntax dances like Slavic, not Germanic. The deeper linguists looked, the stranger it became. Then came a shock, not from linguistics, but from genetics. In 2016, a controversial study by L. Haik and others cross-matched linguistic patterns with genetic mapping. The results suggested that the genetic center of Ashkenazi ancestry was not in Germany at all, but near northeastern Turkey, along ancient Silk Road routes. The language of exile may have been born not in settled cities, but in motion, along trade caravans and merchant roads, places where tongues mixed and identities blurred. Suddenly, the Slavic grammar of Yiddish and its echoes of Iranian and Turkic languages made more sense. They weren't noise, they were fingerprints. Could it be that early Ashkenazi Jews weren't just moving west from Rome, but also north from Persia, east from Byzantium, south from the Khazar steppe? Could a scattered class of Jewish merchants have forged a language outside any single homeland, a code of the road, rather than the echo of one country? If so, Yiddish is not just a dialect. It is a code, a record of forgotten migrations, a living artifact of a multi-directional diaspora, built from Slavic rhythm, Iranian bones, Hebrew heart. And yet, history filed it under German, just as it filed its speakers under Ashkenazi. If the language itself doesn't truly come from Germany, what else about Ashkenazi origins has been misunderstood? To untangle that, we travel to the crossroads of empires, where a Jewish kingdom supposedly rose and then vanished. Could an empire that left no obvious trace still echo in our DNA? This is one of the most controversial puzzles in Jewish history, the Khazar hypothesis. In the 8th Abruzd 10th centuries CE, the Khazars ruled a powerful state between the Black Sea and the Caspian, controlling trade along the Volga-Don corridor. They weren't just warriors and merchants. Multiple historical sources, including Al-Masudi, Yehuda Halevi, and Constantine VII, describe a stunning political shift. Khazar royalty and nobility formally converted to Judaism, possibly around 740 CE. The bold idea that followed: after this conversion, large segments of Khazar society, perhaps tens of thousands, became Jewish, migrated west, and formed the core of the Ashkenazi population. This is the heart of the Khazar hypothesis, brought to global attention by Eran El-Haik's 2013 paper in Genome Biology and Evolution. El-Haik used principal component analysis and admixture models, comparing Ashkenazi Jewish genomes to modern Armenians, Georgians, and Adyghe Caucasians, and found that Ashkenazi Jews clustered closer to the Caucasus than to the Levant. But the backlash was fierce. Leading geneticists, including Doron Bahar, Harry Ostra, and David Goldstein, systematically challenged El-Haik's interpretations. Why? Because while Ashkenazi genomes do show mixed Southern European and Middle Eastern ancestry, they do not show the specific genetic patterns expected from Turkic or Highland Caucasian populations. For example, J1 and J2Y DNA haplogroups, classic Semitic-associated lineages, are dominant in Ashkenazi men. The key maternal lineages, K1A, 1B1A, N1B, and others, trace back to prehistoric Europe, not to the Caucasus. Autosomal analyses place Ashkenazi Jews in a genetic triangle between southern Italy, the Levant, and Iran, but not deep in the Caucasus highlands. So where did El-Haik likely go wrong? Critics argue he used modern Armenians and Georgians as stand-ins for Khazars, even though those populations are genetically distinct and geographically separate from the Turkic steppe elites who likely ruled the Khazar Empire. Still, one detail refuses to disappear. The R1A Y2619 subclade, found in over 50% of Ashkenazi Levites, splits off from other Eastern European R1A about 1,000–1,200 years ago, roughly the same period as the Khazar conversion. Some scholars, including works cited around 2013, have proposed this could reflect a single high-status male founder of Eastern European origin, possibly linked to a Jewish elite in or near the Khazar sphere, whose descendants left a disproportionate legacy. No other known population shares this exact Levite R1A lineage, so maybe the Khazar kingdom didn't father a nation, but it might have fathered a priestly line. And still, the core problem remains. We have no confirmed Khazar skeletons with sequenced DNA, no Khazar cemeteries we can definitively test. Without ancient Khazar genomes, every link is circumstantial. The debate refuses to die not because the evidence is strong, but because the void in the Ashkenazi origin story is so large that even a ghost kingdom can seem tempting. So if not Khazars, and not purely Rhineland Germans, then who were the real Ashkenazi founders? To answer that, we stop chasing empires and start listening to inheritance. Not conquest, birth. Out of thousands of possible ancestors, just 4 women seeded nearly half of the maternal line. You'd expect a people shaped by centuries of diaspora to have hundreds, even thousands, of maternal origins. But when researchers sequenced the mitochondrial DNA of Ashkenazi Jews, they didn't see a vast forest. They saw 4 enormous trees. Costa et al., 2013, and Behar et al., 2006, revealed a staggering truth. Nearly 40% of all Ashkenazi Jews today descend maternally from just 4 women. Their mtDNA haplogroups, K1A1B1A, K1A9, K2A2A1, and N1B, are unusually concentrated, with K1A1B1A alone accounting for about 20% of modern Ashkenazi maternal lines. Here's the twist. None of these haplogroups are Levantine. They do not trace back to the ancient land of Israel. They do not match the maternal DNA of Sephardi or Mizrahi Jews. Instead, they trace back to prehistoric Europe, likely to populations in regions like France, Germany, or the Danube Basin more than 7,500 years ago. This isn't speculation. It comes from deep time coalescence analyses and mtDNA mutation clocks. So how did these ancient European women become the founding mothers of a Jewish people? The most accepted answer: conversion. During the early Roman Empire, between the 1st and 4th centuries CE, Jewish communities were spread across southern and central Europe. Roman historians, including Dio Cassius, record significant waves of conversion to Judaism, especially among women. Those 4 founding mothers were likely part of that story, European by ancestry, Jewish by choice. They married into small diasporic Jewish communities, carried the faith forward, and passed on not just belief, but biology. Through centuries of bottlenecks, persecution, exile, and migration, their mitochondria endured even when their names were long forgotten. 4 women, 40%, an unbroken maternal legacy. They did not write scripture, they did not define Jewish law, but they shaped the bodies that would carry those traditions. So the real question becomes, if Europe gave the mothers, who gave the community its physical soul, what force shaped the survival of this people? The answer doesn't come from legend. It rises from something colder, sharper, buried in medieval bone and sealed in genetic silence. They didn't just survive exile, they passed through a genetic eye of the needle. In genetic terms, this is called a severe founder bottleneck, a moment when an entire population shrinks down to a tiny number of ancestors. For Ashkenazi Jews, this wasn't a metaphor, it was math. By the 13th or early 14th century, models show their total population compressed to just 350 to 500 people, fewer than a small village. From this micropopulation came millions. The Erfurt Cemetery, excavated in 2022, gave us ancient DNA that stunned even seasoned researchers. 27 individuals, buried just before the Black Death, already carried the full genetic profile of today's Ashkenazi Jews. That means the bottleneck had already happened. The genome was already set before the plague arrived. What followed was not comfortable growth, but survival under relentless siege. From 1349 through the 16th century, Europe repeatedly turned on its Jews. Massacres in Mainz, Strasbourg, Erfurt. Expulsions from England, 1290, France, 1306, Spain, 1492, Portugal, 1497. But the DNA held. And that is where the true cost becomes visible. That tiny founder group passed on more than memory. They passed on rare mutations carried silently in the genome, amplified with every generation. Tay-Sachs, Canavan disease, Gaucher disease, BRCA1, BRCA2. These are not just medical terms. They are signatures of a population compressed so tightly that hidden recessive mutations could suddenly rise to the surface. Not destiny, not punishment, just the statistics of small numbers. Yet through all of this, something more than DNA survived: ritual, language, law, identity, passed in whispers, taught in exile, protected in ink and fire. And still, buried in Erfurt, one anomaly refused to fit the model: a single genome with ancestry deeper and harder to classify, hinting at an even older layer of story we still don't fully understand. In that same cemetery, scientists noticed something else. Not one Ashkenazi community, but 2. In 2022, a team from the Max Planck Institute analyzed DNA from 33 individuals buried in the Erfurt Jewish cemetery in the 1300s. The results broke the idea of a single, uniform, medieval Ashkenazi group. Instead, they found 2 clusters. Group A, genetically closer to Western European Jews, with a higher proportion of Levantine ancestry, especially in paternal lines, echoing the remembered Near Eastern homeland. Group B, tilted more toward Eastern and Central Europe, showing stronger affinity with local host populations, likely shaped by admixture with surrounding Christian communities or recent converts. Yet, the shock came here. Despite their differences in genome-wide ancestry, both groups shared the same founder mtDNA haplogroups, K1A1B1A, K1A9, N1B, and both had high frequencies of the same rare disease variants linked to Tay-Sachs and Gaucher. In other words, they had the same maternal roots and the same disease risks locked in before the branches diverged. By the 14th century, the Ashkenazi population was already split into genetically distinct subgroups, yet biologically unified by the same founding mothers and the same bottleneck. Like a single tree with 2 major branches, grown in different soils, fed by the same root. Forced migration and expulsion had brought them together again, to be buried side by side. Science had uncovered two stories in one graveyard, shaped by different histories, fused by blood. And still, the excavation isn't the end. One skull, buried deeper than the rest, chemically distinct, with an even more unexpected genetic signal, still hints at layers of history we haven't fully read yet. Beneath all of this lies one final revelation. DNA remembers, but it cannot decide who we are. For centuries, Ashkenazi identity was written in exile, spoken in Yiddish, guarded in ritual, scarred by survival. Beneath every tradition, a code was quietly carried, a genetic memory that began in the Levant, branched through the Mediterranean, and refracted across Slavic lands. Modern genetics tells us this. Ashkenazi Jews carry paternal haplogroups from the Near East, shared with Palestinians, Bedouins, and Druze. Yet nearly half of their maternal lines trace back to prehistoric European women who probably never heard of ancient Israel. Science revealed something stark. Their fathers prayed facing Jerusalem, but many of their mothers were born before the Torah existed. This isn't a contradiction, it's a fusion. From Roman-era converts in the 1st century to Silk Road trade routes in the 9th, to Erfurt's medieval bottleneck in the 14th, every twist reshaped flesh, not faith. So what is Jewishness? A people? A religion? A genome? Genetics can track mutations. It can map migrations. It can expose forgotten chapters of exile. But it cannot define meaning. Because identity is not an algorithm. It is memory, language, story, ritual. Trauma passed down in whispers, joy hidden in candlelight. The choice to return, again and again, even when scattered across continents. And the map is still incomplete. In southern Italy, excavations are uncovering remains from a forgotten Jewish community expelled in the 16th century. In Toledo, Spain, bones believed to belong to pre-Inquisition Sephardim await sequencing. Somewhere in those remains may lie the true fork between Ashkenazi and Sephardi Jews, the moment the family tree split in two. And maybe, buried in another cemetery, awaits the real key to the mother of all Jewish diasporas.